Any questions, anyone? Your exams will be ready tomorrow. That's one of the things I had to do yesterday. So. Okay, as far as lab is concerned tomorrow, we will continue some more of our staining procedures to gather some more information about your unknown. Tomorrow will be some important ones for those of you that have gram-positive bacillus unknowns. We'll be doing the endospore test. Go ahead and leave your sporulating agar tubes in the incubator. We want to make the conditions as bad as we can to try to force cells into producing endospores if they can. So just leave those in there. For the capsule stain, you don't really need a culture of any particular age. So as long as you have some viable growth on the slants, you can do the capsule stain. And then we're going to begin an exercise called a peer culture exercise. In that particular exercise, each one of you is going to be given a tube of somewhere between 2 and 4 organisms in a broth. And your job is to tell me not what they are, but how many different organisms are in your sample. So the answer will be somewhere between 2 and 4. And then you're going to have to justify why you believe that. Basically, this is a way to apply the streak plating techniques that you've been practicing now, and also gram staining. And so this is a practical application of those. This will be an exercise that you can work on for the next week. It's not going to be due immediately. Typically, I don't make this due for a couple of weeks, but it's something that allows you to kind of pull some of the things together that we've been working on in lab up to this point. Okay? I updated the stuff on Moodle so that there are now a set of PowerPoints and study guide questions and eventually lecture notes, which will reflect the part of the material now which is exam 2 eligible. So we're still talking about growth of bacteria, but on those particular materials on Moodle it'll say growth exam 2, for example. So those are the ones that you want to be looking at. Okay, so last exam, we talked about microbial growth in the context of looking at how populations changed over time and looking at growth curves, and then also ways in which we could count the number of organisms in a population to generate growth curves. Now I want to look at the other side of things and take a look at some of the factors that influence microbial growth. And we're going to concentrate on a few environmental variables that are quite important, one of them being temperature. Obviously, one of the things we had determined early about your unknown was what temperature does it grow best at, which incubator should you put it in. Another factor that we'll talk about will be pH. We haven't really dealt with pH much. That's kind of taken care of you for your unknown, because when media is prepared for you, that media is buffered to assure that it is a particular pH. But we'll talk a little bit about the influence that pH can have on growth. Getting the proper food resources is obviously important to sustain growth. So we'll talk a little bit about the composition of media and the kind of things that bacteria absolutely need to have to be able to grow in a laboratory environment. And then we'll talk about some gases that influence microbial growth, and particularly we will concentrate on oxygen. And that's something that will lead into our next topic, which is the topic of metabolism, and then that will also lead into a lab we'll do in a couple of weeks, where we look at and investigate what are the oxygen requirements of your unknown. How can we categorize it based upon the type of oxygen that it needs, or the type of respiration or fermentation, for example, that it's going to be able to carry out. So gases are really pretty important. So let's take a look at temperature first. Temperature is pretty straightforward. Oops. So if we make a plot of rate of growth of a microbial population versus temperature, what we can see is that there are three kind of general patterns that define how microorganisms grow in response to temperature. The blue pattern, the green pattern, and the red pattern. Psychrophils, mesophils, and thermophils. When we talk about temperature profiles, there's typically three things that you concentrate on. And one of the things you concentrate on is, what is the minimum temperature that organisms of that particular category can grow? Likewise, what is the maximum temperature that they can grow at? And finally, what is the optimum temperature? You can see that in these three particular cases, there's a fairly distinct demarcation between optimum temperatures and actually very little overlap between the maximum temperature of one group and the minimum temperature of the next group. Now, that isn't always true, because one of the things that we're going to see is that depending upon the species of microorganism we're talking about, these curves can be very narrow or they can be very broad and spread out, depending upon the species. So, for example, I think that I might have mentioned before that Mycobacterium leprae, the bacterium that causes leprosy, has a very narrow temperature optimum. It's somewhere between about 33 and 35 degrees Celsius. And that's a temperature that sometimes is most common in the extremities of the body, like your fingers, your toes, maybe your face. And those are the kind of regions that the leprosy bacteria therefore tend to colonize. Organisms tend to live in places where their temperature needs are met. Okay? But a very narrow temperature range. Okay? On the other hand, Staphylococcus species like Staph aureus and Staph epidermidis, which grows on your skin, can have a huge temperature range, going from like 6 degrees Celsius up into the 40 degrees Celsius. So it's got a very broad, spread out temperature profile. And so these don't reflect the temperature profiles of all bacteria. This is just a representative example of some of those. So let's take a look at psychrophils for a second and just work our way left to right. You don't have to memorize these numbers, and so I don't expect you to know that typically speaking, the minimum temperature for a psychrophil is about minus 15 Celsius. The maximum is about between 10 and 15, and the optimum is right around 0. But you should understand that these are the cold-loving organisms. Okay, so they tend to prefer colder temperatures. Okay, so what are some of the implications for that? Well, some of the implications for that are that we typically do what with our cultures once we've incubated them? We refrigerate them. Why? Well, there's a couple of reasons. Number one, we want to slow down their metabolism so that they will last longer. But also in the refrigerator, if we slow down their metabolism, if there are any contaminants, then we slow their metabolism down too. So a way to kind of preserve your cultures and try to make sure they don't get contaminated or the contaminants don't grow is to refrigerate them. We use that strategy in trying to preserve our food. Okay? Food out of the refrigerator obviously spoils faster than food in the refrigerator, because most of those agents of spoilage tend to be these mesophilic organisms that have a higher optimum temperature. Okay, room temperature is typically around, between 20 and 25 degrees Celsius. And if you pop it in a 0 degree refrigerator, that's going to significantly retard their growth, unless those organisms are psychrophilic, in which case you've got them at their temperature optimum. And so that's why some things can spoil, even if they're refrigerated, even in relatively short periods of time, because the spoiling organisms are these type of cold-loving bacteria. Psychrophilic organisms can be found in a number of environments besides refrigerators. They can be found deep in the ocean, where temperature decreases as you go deeper and deeper into the water. They can be found in snow, in polar ice caps, again showing the adaptability of microorganisms to survive in some pretty extreme type environments. There are some organisms that deserve mention. And those are organisms called facultative psychrophils. We're going to see this term facultative later on when we talk about respiration. Facultative psychrophils are actually more commonly, in terms of their category, a mesophil, one of these middle type of organisms. But what the facultative indicates is that they are able to tolerate the colder temperatures. It's not optimum for them, but they can survive in those lower temperatures even though it's not their preference. So some of these facultative psychrophils can also contribute to spoilage in the refrigerator. They'll grow at the lower temperatures, but they do so extremely slowly. Okay, a true psychrophil would grow readily at those lower temperatures because that's its optimum. Facultative psychrophils can tolerate these temperatures. They don't kill them, but they grow much, much more slowly. Mesophils fall into this, typically around 20 to 40 degrees Celsius temperature. This is where most of your organisms you deal with fall into, because our major temperature incubations are 25, 30, and 37. Probably the biggest significance that we'll encounter in terms of mesophils will be the fact that human pathogens fall within this particular category. Why? Because internal body temperature is 37 degrees Celsius. There are some mesophils that are of concern to us, and these are some mesophilic organisms that are said to be thermoduric. And these are kind of the warm equivalent of the facultative psychrophil. Thermoduric can tolerate temporarily higher temperatures than this optimum and profile would suggest. Now the key is they can't tolerate it very long, but they can tolerate some short periods of heating. When you're cooking materials that could be contaminated with pathogenic thermoduric organisms, you've got to be careful that you cook the food to the proper internal temperature and maintain that temperature long enough to kill even these thermoduric organisms. One of the types of foods that's commonly contaminated with some nasty intestinal pathogens, like the hemorrhagic form of E. coli K12, is meat, okay? And particularly ground meat, which could have been contaminated with intestinal material from the cow that's been slaughtered, for example. In this particular situation, if you've got these E. coli thermoduric bacteria, even if you temporarily heat them up past what normally would be their maximum, many of them can survive. And then when the meat cools down again and you ingest them, they are still viable. And so that's one of the reasons why there are public health standards for cooking meat to proper internal temperatures, specifically to deal with these particular type of thermoduric pathogens. Thermophils are pretty interesting. Thermophils have a very high temperature optimum, a temperature optimum that would normally kill most organisms. What's the major effect of temperature from a negative standpoint of killing microorganisms? What's the mechanism? Why does heating a microorganism tend to kill it?
Because the proteins do it.
Yeah, denaturing proteins. Okay. Proteins unfold, remember, and hydrogen bond breaks as temperatures increase. And so these thermophilic organisms, which can tolerate boiling hot springs, they can tolerate temperatures of incredible proportions in these areas called undersea vents, where the magma of the earth under the ocean has split and superheated water is coming out of those fissures, and because of the pressure of the water, the boiling point is elevated, and water can get to be like 256 degrees Celsius down in those thermal vents. And there are bacteria living down there. Okay? So one of the questions becomes, what kind of molecular adaptations do they have to try to prevent their enzymes from denaturing? Okay? In these thermophils, if you cool them down to what normally would be a mesophilic region, typically they're not very viable. One of the benefits that lower temperatures have is that we tend to get an increased rate of metabolism as temperature increases. And typically for every 10 degrees Celsius that you increase temperature, there's typically about a doubling of metabolic rate. It's called Q10. And that persists until you exceed the temperature maximum of that particular organism and enzymes start falling apart. So temperature is an interesting phenomenon. Increases in temperature within this range can speed up metabolism. But if you get it too hot, then you start denaturing proteins and killing the microorganisms. Now, you get a similar response to pH if you plot rate of growth versus temperature. And I'm not sure if I have a pH curve or not. I don't think I do. Oh, here's some psychrophilic bacteria growing in snow. They're forming this particular red pigment, which is a photosynthetic pigment. So these are some of the coal-loving bacteria. Here's what these particular bacteria look like. Oops. So let's go back to this curve for a second. So if we substituted pH for temperature, typically, again, for a given organism, there will be a minimum pH, there will be a maximum pH, and there will be a pH optimum. Remember, this is similar to enzymes. Enzymes have that minimum, maximum, optimum. And so one of the big effects that pH has on growth is it affects enzymes much like temperature does. Remember back in cell biology when you did your turnip peroxidase project, one of the things you were trying to determine was what is the pH optimum of your enzyme and what is the temperature optimum of your enzyme. Different organisms have different pH optimum, and they range all over the pH scale, from as low as 1 or 2 to as high as 12, 13. Normally, we would think that those particular extremes would be deadly, but again, organisms have adapted to tolerate certain types of pHs, and that's allowed them to colonize particular environments that other organisms can't tolerate. So, for example, there is a bacterium called thermoplasma, and these particular bacteria not only can tolerate higher temperatures, but they can tolerate very, very low pHs, down in the region of like 2. One of the places they tend to live is in old coal slag piles. And coal slag piles typically have a lot of sulfur associated with them, and the sulfur dissolves in the water from the rain and creates sulfuric acid. And so those coal piles are very, very acidic, and yet these particular bacteria thrive in those particular environments. Generally speaking, fungi tend to have a little bit more acidic pH optimum. Bacteria tend to be happier as a rule in slightly more neutral type of pHs. Consequently, when our tryptic soy agar and tryptic soy broth is made, that is buffered typically to be at a pH of between 7 and 7.5. That accomplishes a couple of things. Number one, it keeps the pH of the media in the sweet spot for most bacteria. But number two, it tends to discourage the growth of any fungal contaminants that might have gotten into that plate or that media that weren't destroyed by autoclaving. Now, there are some fungi that are adaptive and can tolerate those higher pHs. When you did your plates, when you intentionally exposed your plates, one of the things you oftentimes saw were fungal contaminants that had this kind of cottony appearance. All those fungal hyphae give it that appearance. And those were bacteria that were able to tolerate that more neutral, basic type of pH. Another factor that can influence microbial growth is solute concentration. I've already mentioned the fact that one of the early ways that was used to try to preserve food was to do things like salt it. Salting creates a hypertonic environment and would draw the water out of bacterial cells that might be contaminating that food. There are some highly osmotolerant organisms, and we mentioned the fact that the RK bacteria, some of them were very osmotolerant and could live in environments like the Dead Sea, the Great Salt Lake. One of the ways they can accomplish that is that they are able to actually pump solute from their environment into their cytoplasm, and in doing so, create a cytoplasm that is isotonic to that normally very salty environment. So if you do an analysis of a salt-tolerant bacteria versus a non-salt-tolerant bacteria, the salt-tolerant one would have a much higher osmotic concentration of solutes in their cytoplasm. Again, there is a real range of salt tolerance exhibited by different species of microorganisms. One of the things we will do in lab is we will plate out both known organisms and your unknown on different agar plates with different amount of added salt to the environment. And one of the things you'll do is say, will it grow on these salty plates or will it not grow on those salty plates? One of the things that you'll find is that some bacteria are highly tolerant to solute and others not so much. A couple of genera that tend to be a little bit more salt tolerant are especially Staphylococcus and to a lesser degree Bacillus. So we'll have some agar plates, for example, to which 15% added sodium chloride has been added. For most of your unknowns, they won't grow at all. That high solute concentration will basically suck the water out of them and kill them. But some will grow quite well, particularly the staph. That's adaptive for certain species like Staph epidermidis, which live on your skin. Why? Because your skin tends to be a salty environment because of the evaporation of the liquid, sweat, and leaving behind the solutes that were in that sweat, creating a fairly hypertonic environment in your skin. And that staph epidermidis can tolerate that environment and actually thrive under those conditions where many other bacteria really can't. And then finally, another environmental condition that's pretty important is making sure that there is sufficient water available to sustain growth. Remember that the cytoplasm consists of about 70% water. And so it's important that the cells be constantly hydrated. If they become dehydrated, metabolism slows, and the cells will eventually die. Now, there are a number of other nutritional factors that have to be provided in addition to these environmental factors that we've been talking about. So what kind of nutrients do microorganisms need to grow? Well, if you do an analysis of a typical E. coli cell and start breaking it down, you see that there are some molecules that are quite important. About 50% of the dry weight of the E. coli is protein. About 20% of the E. coli is RNA. Smaller amounts of other materials, lipids, miscellaneous compounds. If you start looking at some of the elements that make up these particular molecules, you'll see that E. coli is mostly carbon, oxygen, and nitrogen, hydrogen, and then a little bit of phosphorus, sulfur, potassium, sodium. And then there are some other elements that typically are called trace elements that represent smaller amounts of needed substances. So when you start thinking about nutritional needs, you have to ask yourself, where is that bacterium getting from its food supply its carbon? And where is it getting its hydrogen and its oxygen and its nitrogen? Now, some of those things are coming from the environment. Some of those are gaseous materials. But other things, like the carbon particularly, come from the food it eats. So we've made media, and we have made both solid media and we've made liquid media. When you look at media, some of the big questions you have to ask is, how is it providing carbon and nitrogen for the organism's growth? Another question you have to ask is, how is it providing energy to sustain the growth of that microorganism? Does anybody remember what we mixed together to make broth in the first lab? All those little bottles that were over there on the side that you poured together and put in water and dissolved. Do you remember anything that was in broth? There was something called peptone. And basically peptone is digested protein. The source of the protein can vary. One of the media that we use most typically is something called tryptic soy agar. Tryptic refers to the fact that the enzyme trypsin is digesting the soy protein. And what is it producing for the bacteria when it does that? A source of carbon, a source of nitrogen, and those are needed for making new molecules. carbon and nitrogen are needed for biosynthesis. Okay? All organic molecules are carbon-based, right? Proteins, nucleic acids, carbohydrates, lipids. And many of them contain nitrogen as well. Nucleic acids contain nitrogen. Amino acids contain nitrogen. So we need a source of carbon and nitrogen to make new molecules so organisms can grow and reproduce. Additionally, these digested amino acids can be used as a source of energy to make ATP to support cellular activities. One of the cellular activities that's supported is biosynthesis, making new molecules. So this peptone is really important in terms of a nutrient source to sustain microbial growth. Another thing that we put into that particular broth was something called beef extract. And beef extract is a source of several types of substances, including some important vitamins and minerals. And on this right hand of the list over here, for example, some of the important minerals that you find in beef extract include things like iron and chlorine and calcium and magnesium. Sometimes these minerals are referred to as micronutrients. It's not because the minerals are small, it's because these things are needed in small amounts. You don't need a whole lot of them in your diet. The beef extract we used to use in that exercise until about 5 years ago was this nasty, foul-smelling, brown, pasty stuff that was made by just taking beef and boiling it down and letting it get soft and mushy. And that's what you would scoop into your flasks to make broth. We now have a dehydrated beef extract, which is much easier to work with and much more palatable. But that's providing mainly these micronutrients, vitamins, minerals, and things. You threw a little sodium chloride in there so you could make it isotonic. You dissolved all of this up in water. Obviously, microorganisms require liquid in their environment. And you got to dissolve this stuff. And we didn't do it, but it probably wouldn't be a bad idea when you're making your broth to go ahead and check the pH and make sure it's in that sweet spot of 7 to 7.5. So that's broth. What did we do when we wanted to make the auger then? We took some of this leftover broth and we just add a solidifying agent, auger, to it to make a plate or a slant. An amazing number of bacteria can grow on this tryptic soy auger or tryptic soy broth. And it's really pretty remarkable when you think about it. Because this is the only source of food, basically, that you're providing these things. I could lock you in a room and for the rest of your life just feed you agar. You wouldn't last very long. Your cells do not have the metabolic capability to take these basic substances and turn them into all of these particular things that you need for cellular metabolism and growth. But bacteria can do it. One of the themes that I'm going to reiterate to you over and over again is that while bacteria are very simple structurally, especially compared to complex eukaryotic cells, metabolically they're very sophisticated. And they really put our cells to shame. Metabolically, bacteria can do things we could never do. They have a lot more capabilities in terms of taking basic materials and turning those into anything the bacteria needs to grow. Eukaryotic cells have much more limited metabolic capabilities. Organisms that can live on these two types of basic media, and these two particular types of media are called minimal media, are pretty common. All of your unknowns, for example, do very well on these basic minimal media. But there are some organisms that won't do so well growing on minimal media. These organisms are said to be fastidious. They have special requirements. It might be a particular growth factor that isn't present in the normal medium. It might be a special vitamin that isn't present in that beef extract, for example. We're going to use a media later in the semester that is designed to encourage the growth of some fastidious pathogens. And that particular type of media is called blood agar. Blood agar is made by just taking this tryptic soy agar and then adding sheep blood to it. And so the plates have this nice, pretty looking red color. And one of the things you're going to do with blood agar is you're going to take throat swabs and you're going to streak out throat swab material from your body onto this blood agar and see if you can get some pathogens to grow. We're going to see if you're a carrier for some of these pathogens. Why do these organisms need this blood to grow? Well, they're not vampire organisms, technically, but they require the heme portion of the blood, which specifically is the iron-containing portion, and that is one of the micronutrients that they tend to need higher levels of. When we talk about metabolism and later on the body's defense system against growth, one of the things we're going to see is that in terms of essential nutrients, iron is really important to microbial growth. And if you limit iron availability to microorganisms, that can really retard their growth. And that's what some of our defense mechanisms against pathogens tend to do, is they tend to make iron unavailable for bacterial growth. So we'll come back and talk about that a little bit later on. Well, there are some gases which are incredibly important in determining microbial growth. The two gases that we will talk about most will be oxygen and, to a lesser extent, carbon dioxide. When we talk about oxygen, we need to delve into a concept that's a little confusing at first, but is very, very much important in determining what an organism's oxygen requirements are. And that concept is the concept of an ROS. ROS stands for reactive oxygen species. ROSs are unusual forms of oxygen. They are ions or free radicals. And one of the things that these reactive oxygen species can do is destroy cellular components. For example, ROSs can oxidize cell membranes, and that's not good. They can oxidize and cause damage to DNA molecules, and that's not good. So basically, these particular substances tend to be toxins. So where do ROSs come from? What's their source? Well, there are some natural sources of ROSs. Oxygen can interact with sunlight, and chemical reactions can be initiated, which generate some of these reactive oxygen species. And typically, the more oxygen that's present and the more sun that's present, the more ROSs that are present. Another important source of ROSs is aerobic cellular respiration. And particularly in the electron transport chain. Remember in the electron transport chain that the purpose of the electron transport chain is to recycle your reduced coenzymes? Remember that from cell biology? And as a result, a hydrogen ion gradient is formed as the electrons pass through the electron transport chain, and ATP is made by chemiosmosis. And what eventually happens to those electrons that pass through the electron transport chain? They're picked up by oxygen and used to make water. This represents a complete reduction of oxygen, formation of water. But sometimes, many times, the reduction of oxygen is incomplete, and we also then can produce these toxic byproducts, these ROSs, these reactive oxygen species. So really, these toxins are kind of a natural byproduct of this important process of aerobic cellular respiration. It's almost kind of like the price a cell makes for having this really efficient way of producing ATP. Okay? So it's the dark side of cellular respiration, if you like. So, these are going to be produced, okay, either in the environment or by cellular respiration. So how does the cell tolerate these particular things? Well, one of the things it can do is that it might have mechanisms that can detoxify some of these ROSs. And there are enzymes that can do that. There are some chemical compounds in the cell that can scavenge or tie up these ROS compounds and inactivate them. Those particular compounds are called antioxidants. And Dr. Morgan McCombs does research with antioxidants in yeast and ROS species. If the ROSs come from natural sources, like the effect of sunlight on oxygen, then there are some pigments the bacteria can do that can also scavenge or tie up those ROSs. Many of these pigments are the yellow-orange pigments. Those yellow-orange pigments are the carotenoid pigments. So some of the bacteria that you see that produce these yellowish-orange pigments, that's one of the purposes of that particular pigment, is to help protect the cell from some of these damaging ROS species. You've already encountered, way back in cell biology, an enzyme system which is responsible for detoxifying a particular ROS. What was that? That was the turnip peroxidase enzyme, right? And what did that peroxidase enzyme do? It broke down the reactive oxygen species, hydrogen peroxide, which is a toxic compound. Why was that peroxide being generated? It was being generated as an unwanted byproduct of those cells' aerobic cellular respiration. So, what we're going to do next time is look at a few of these ROSs and see how they're produced and then what mechanisms these organisms have to deal with the presence of these damaging ROS species, and tie that back to then the types of environments organisms can live in and why some can tolerate those environments and other organisms can't. And that will lead into a lab we'll do in a couple of weeks. So, again tomorrow, capsule stain, endospore staining, and we'll start our pure culture exercise.